Developing robust cell harvest and media exchange processes becomes more challenging as programs move from development into GMP manufacturing. Differences in scale, equipment, and automation can introduce variability, complicate tech transfer, and delay clinical timelines, while conventional approaches such as TFF and disk stack centrifugation face limitations around scalability, shear, and process comparability. Today, we'll hear from Mark Smith, Senior Director of Innovations and Applications at CARR Biosystems, who will share case studies illustrating how a low-shear, single-use centrifugation approach enables efficient harvest and media exchange across cell types, including iPSC aggregates and MSCs. This data will demonstrate how consistent process parameters can be maintained from development through commercial manufacturing, preserving critical quality attributes while reducing engineering complexity and shortening time to clinic. I'm your host, James Crosspayne with Cell and Gene. I'd like to thank you, the audience, for attending today's event. This is live, so we do encourage your participation. Take a moment to get comfortable with the tools on your screen. Our presenters have shared a few resources for additional learning below the slides. To the right, you'll see Mark's contact info, and just below me, a question box. Please share your thoughts with us during the presentation. We will answer as many questions as possible, and any we miss, I'll pass along for follow-up after the event. This question box can connect you with me if you run into any trouble, but please try refreshing your browser first, as it will fix most common issues. Finally, today's event will be available on demand. You'll receive an email with a shareable link shortly after we conclude. And also, our presenters have asked a few questions of you to the right of the slides. Please take a moment to share your feedback with us. Now, without further ado, let's begin today's presentation. Welcome, Mark.
Well, thank you for the introduction, James. It's my pleasure to be here today to talk to everyone about the simplified harvest and media exchange and how you can handle your cell processes. Well, we want to start out today with a bit of a poll here and understanding how do you, the audience here, process your cells to achieve optimal densities of your cells and in the desired solution throughout the workflow. And so while you answer that, I'll give a little bit of a background of what we're going to be talking about today and a little bit of background on CARR Biosystems. So for today's agenda, we'll be talking about our simple scalable cell processing. We'll give, again, that brief background from CARR, talk about some typical cell processing technologies. Then we'll take a deeper dive into our unifuge system for scalability and performance. At the end of that, we'll hit a couple of key case studies there, iPSC, differentiation, passaging, and MSC cell harvest, and then we'll wrap for today. But who we are as CARR Biosystems, we're driven to advance life-saving and life-enhancing solutions. We're part of a greater company called Berry-Waymiller, which is over a $3 billion company in revenue, over 12,000 people globally. CARR Biosystems was founded back in 1993, and while over the years we've rebranded, we've built a large global reach with a US manufacturing center. And critically, we have technologies that play essential roles across multiple FDA and globally licensed products and processes. And the products we've developed over our 30 years of innovation include our clean-in-place and steam-in-place equipment, or often referred to as our stainless steel equipment, which includes our PowerFuge family, which is a high G-force centrifugal separator, typically used in vaccines and microbial production. ViaFuge, which is a gentler separator that can recover, which is excellent at clarification, but also cell recovery. And today we'll be talking mainly about our single-use centrifuges, or our unifuge family, which has really low shear, excellent clarification, and is very good for live cell recovery. And when we look at the different modalities we support with these technologies, it spans the gambit here from cell therapies, gene therapies, biologics, mAbs, vaccines, and cellular agriculture. Of course, today we'll really focus on the cell therapy aspects of it. As we focus on the cell therapy aspects and look specifically at the unifuge family, we have a number of places and use cases we typically see our technologies being used across harvest, washing the cells, buffer and media exchange throughout the workflow and the process, concentrating the cells, and formulating and pre-formulating for final fill and finish. So let's review those poll questions. where are you today? How do you achieve optimal densities and desired solutions? It's interesting to see, and maybe not surprising to see, that a lot of us are in manual kind of quasi-closed systems with maybe using biosafety cabinets. We'll talk a little bit more later in the slides here about why that might be and why there might be some interesting choices to make in terms of ensuring future success. So in terms of cell handling and processing, I'm going to use these terms a lot, and so I wanted to get down into what is cell handling and what is cell processing. And unlike a single unit operation, where the unit might expand the cells, that's a bioreactor, or might electroporate, transduce the cells. Cell handling can be the critical manipulations that bridge the gap between unit operations or between process steps in your workflow. But typically they break down into two main steps. That's media exchange or buffer exchange, where the cells are retained and the solutions exchanged. And that could be, for example, spent media going to fresh media, or that could be exchanging a buffer for a critical chemical or hormone to induce differentiation. The other side of things is cell concentrating, right? The cells are retained and the solution is drained. And with these two devices, we see that frequently through the workflow. These kind of summarize many of the cell handling and cell processing steps that happen and bridge between the unit operations. So with that, how do you handle, I believe this is coming up. Excuse me, I'm out of source here. Where do you see these in the workflow? Really all across the workflow here. We see leukopak washing, for example, in buffer media exchange, where you may want to wash out cryo buffer or other elements in the leukopak. Pre and post selection of where you might want to be adding reagents or removing reagents. Cell differentiation, adding cytokines and hormones or removing them is also just as critical. Formulation, getting into the right formulation buffers. Those are a couple of examples of where you might find media exchange across the workflow. And on the cell concentration side, that might be pre or post selection, making sure your your cells are at an optimal concentration or an optimal density to pair with your selection medium. Gene delivery. It might be critical to have the right concentration of cells to your viral vector or cells to your electroporation device. And of course, harvest and formulation, where you want to get to your cells to that final formulation for target dosage and target density. So with that, I'd love to get your feedback on which cell handling process steps are the most challenging for you to get right. Is that media exchange, cell concentration, harvest, cell washing, pre-formulation? And as you answer that question, I'm just going to move forward a little bit. Nothing earth-shattering here, so please do answer. But as you think about a cell therapy workflow, of course, they're all flexible and slightly different. Where do these handling steps come in? From donor and collection, to enrichment, gene modification, expanding into banks, expanding and differentiating, and final harvest and formulation. Across this workflow, where do we see cell handling processes optimize the individual unit operations? Well, from our experience, we see lots of places where the cell handling, where media buffer exchanges as well as concentration, can really optimize the performance and the repeatability of a given unit operation or process. And so really across everything, there's a great opportunity to have a good technology to enable your unit operations. With that, we'll go, hey, which cell processing step is the most challenging for you to get right? Oh, this is very interesting that across the board, we kind of see a lot of different, a good spread across the board. Maybe that's indication that across the board, there's a lot of different challenges here. When you're considering your technology to select, like what technology should you be using for your particular challenge, there's a lot of different ways that we've seen users approach technology selection. And I've kind of broken it into 3 groups here. But the first way is thinking about the technology's accessibility. Hey, do I have access to the technology that enables the cell handling I need, that enables the media exchange? Is it easy to use? Like, does my Like, do I or do my teammates know how to use this and have experiences with this? And can it scale down to the volumes that I'm doing in my process development scale? The kind of a next step is, does this unit operation give me the yield that I want? Does it give me the repeatability that I want? Does it give me the speed that I want? And does it give me process controls that enable that repeatability? Kind of a next grouping would be more strategic. Does this fit in my workflow? How does it fit if I was going to go to manufacturing? How robustly does it scale up, particularly for allogeneic customers, for end users? And how easy can I tech transfer this such that when we go to a GMP facility, we can do this with ease? And so this is just an example set of how you might consider technologies. But if you look across, the reason why I've binned them this way is often when we're early on in drug development or therapy development, we think with entry-level thinking, hey, is it available in my lab? Is it easy to access? But eventually, as we go down this path, we want to be optimizing it for yield and getting it ready for manufacturing. Because ultimately, if the goal is to be able to achieve CMC, we want to have a good strategy to approach there, to be able to deliver robust and characterized manufacturing processes and define and control our CQAs, or critical quality attributes of the product, by defining and controlling our critical process parameters, or CPPs. And the earlier you can do this, control your CPPs and CQAs, of course, the more confidence you can have in your scale up. And if you can get these right the first time when you're doing your process development, then when you get down the road later, when you get towards manufacturing, you can really focus on optimization rather than late stage development of your process. So we'll take these considerations, and like a particularly special consideration to think about is scale up. Of course, scale up has really good value as you're getting to that commercial scale. You need that yield. You want to minimize process lots, minimize the QA, QC sampling and testing, get those economies of scale and labor you're looking for. So one consideration when you're looking to scale up technologies is, are we going to have equivalent controls? Are those controls going to enable us to have the same critical process parameters and have comparable processing times, for example? One thing that's often, or that can be overlooked when you're looking for scale up, though, is scale down. You can't scale up from something that doesn't match your scale, that you don't have a scale down model for. And so the value of having a good scale down model early is that you can do process development and early optimization of your critical process parameters. And if your scale down model is robust, then it enables you to consider those critical considerations like CPPs and controls early in your process steps, early in your process development, instead of having to wait until scale up to get that critical control. So as we think about, the earlier the process you can get the scale down model in place, the more confidence you can have in that future scale up model. And when evaluating overall cell processing and cell handling technologies, there's a couple of considerations we've frequently seen here, and we'll use these considerations later as we kind of give a scorecard to different types of technologies and different types of approaches. So one is ease of entry here on the top left. Is the technology available? Do I have it in my lab today? Do I need to purchase something special? Of course, that plays a role, but we kind of saw earlier in CMC strategy, that may not be the most critical one if we are looking to the future. Performance plays a role. Can it do what I want it to do? And what's the yield? What's the viability? And what are other CQAs there that I can get? Another consideration is riskiness. So in the future, how much risk am I introducing to a process if I choose a technology today? Is it an open or closed process? And I think I saw from our poll question earlier, a lot of the audience today is using kind of a quasi-closed system with biosafety cabinets. So maybe some extra risk there that could be introduced. And the other piece of the risk is, are there CPP, critical process parameter sensitivities? For example, if I change my parameters slightly, do I have a dramatic increase in the time of my process? Or if I increase the time of my process, how sensitive are my cells and my CQAs to those outcomes? Going to the top right here, repeatability. Is this manual or automated? Do I have operator dependence? Is this plug and play every time, or do I have to optimize it every time? And how broad are my operating windows? Again, close to the riskiness of the CPP sensitivities. How broad is my repeatability range? And of course, scale down and scale up. As we talked about here, can I get a predictable process early? Can I look at those controls early? Can I be cost effective early, such that in the future I can be optimizing instead of doing development on my scale up? So with that in mind, I want to take the next little while to talk about a couple of cell processing technologies that we typically run into. So first, I'll ask the question, and I'll leave this up for a minute. What technologies are you currently using for your cell processing across here? Now, I'll move forward. As you answer that, this is just kind of a rundown of those on the list. Of course, gravity sedimentation is one we've seen end users using, where they simply settle out the cells. Benchtop centrifugation seems to be very popular, of course, very ubiquitous. Disk stack centrifuges, we've heard rumblings of, really haven't seen, but it's interesting technology. Spinning membrane filtration, TFF, and of course, tubular bowl centrifuge. So we'll walk through these technologies and kind of rate them again on our experience with end users about how do they fit into overall evaluation considerations for development technology. So looking at your answers, yeah, that's not too surprising to see benchtop centrifugation is pretty popular. That kind of is that quasi-closed system where you're utilizing the biosafety cabinet to kind of give you that closed system processing. And maybe not surprising, ATF, TFF is a strong second here. And we'll talk about that a little bit more here in some of the discussions today. So let's jump in and kind of walk through these at a glance, these different technologies and some of the considerations around them as you're scaling up. So gravity sedimentation, fairly self-explanatory, but this is settling the cells inside the vessel. You're stopping agitation, you're stopping aeration, you're decanting the vessel. So some really good pros in this regard. Simple, it's in the vessel, you have existing equipment, and it can be typically done fairly straightforward in a closed system manner. But there are some challenges with this, whether or not your system has the automation to enable this. Another piece of this is that often dip tubes or other devices to draw off or decant the solution are not necessarily built to be optimized for controlling your cell concentration that you get. And that can be then a challenge for yield and repeatability. But ultimately, one of the biggest challenges we've seen with end users here is scale up. Because in this top right graph, you can see at small volumes, 1-2 liters or less, you might be at 10 minutes to get your 10 minutes of settling time to get your cells to settle out and to exchange your medium. But as you scale up, even to maybe on the order of 10 liters or less, you're starting to look at 1 hour of processing time, 50 liters, you're really starting to stretch that out. So it's very difficult to then kind of get that scalability of the time, the processing time, if that's a critical process parameter for you. So overall, if we looked at this from a quick scorecard, really easy to use, but from a scale-up perspective, and maybe from a process time perspective, not the most ideal approach. Of course, benchtop centrifugation, which we saw was a big winner here in terms of use. Of course, a big part of that is it's ubiquitous. Everyone has a centrifuge. So it's functionally existing equipment for everyone. Simple, typically very rapid, particularly at small scales. Some limitations or challenges that you can run into, though, is this is a very manual process, particularly from the filling and then the decanting process, as well as the resuspension process. And when you're scaling this approach, you have to have more bottles for more volume, right? And so you either have to massively parallelize, have 30, 10, 20, 30 centrifuges, or 3, 5, 7, I don't know what the number is exactly, depending on your scale. But you have to parallelize that, or you have to take a long time and sequentially be operating your bottles. And what that means is lots of bottles being opened and closed, and also lots of labor opening and closing those bottles. And again, if you think about every opening of a bottle, even if it's in a biosafety cabinet, that's a risk, right? You're opening your process. And so one way to think about that risk is in this top right graph. As you process more volume, you're going to open a bottle more often. And the more often you open a bottle, the more risk you have, even inside of a biosafety cabinet. So we've got two different lines here in blue, or the bottom line, we've got concentration. Let's just say you had one concentration step where you're opening the bottle, filling it, closing it, spinning it, opening it, decanting, and then maybe resuspending. Even at 1 liter, you're only at like 2 or 3 bottle openings there. Fine, that's no problem. But by the time you get to 10 liters, you're almost at 30 times the bottle opening. And if you got to 35 liters, basically you're at 100 times you're opening the bottle. That's what that bottom right arrow is. Now imagine through your workflow that twice in your workflow, let's say during your Lucapac or early in your workflow where you're clarifying something or preparing it for gene transfer, and late in your workflow where you're doing formulation, you're going to be doing this type of process. Even when you start getting 2, 3, 4 liters, you're starting to do this tens of times or even hundreds of times where you're opening and closing a bottle. With every bottle opening and closing, presenting a risk, just makes it so the process risk is really high and scale up, at least in our view, is really untenable. Really easy to use, but challenging in the future. So now disc stack centrifugation. We bring this one in the mix, not because we've seen it, but because we've heard, traditionally, of course, large-scale MAPS clarification uses disc stack centrifuges, really robust clarification, and can get to very large scales in terms of processing volume. One of the things that's typically associated with disc stack centrifugation is shear stress. And of course, shear stress can drive to cell death. And cell death when the cells are your product, of course, is unacceptable. The other piece is, if that's not bad enough, I suppose to say, the other piece to say is scale down models. The scale down on disc stack centrifuges really can't scale down beyond the processing minimum volume around 10 liters, making it difficult to have early entry and good process development early on small scales. And if as you go out and people trying to match and scale down these models, you can see the only way they can really scale them down is into benchtop centrifuges. These are some example academic journals trying to do that. And as they scale down into the benchtop centrifuges, the only way they can mimic the performance is by shearing the sample, shearing the cells, which again, is not a great idea for cell therapy when you're trying to get the cell down. So from an ease of entry, it's a bit too big to scale down to, and from a performance standpoint, shear stress and cell death is certainly not ideal. Next up, spinning membrane filtration. I think many of us are probably familiar with this, but if not, this is a diafiltration TFF-like membrane, which utilizes a spinning membrane to create kind of that reduced or that shear effect across the membrane to not have dead-end filtration. And so this has some great pros. This is typically a closed system, can be highly automated, and gives good small-scale performance. And from some end users that we've worked with, one of the things that we've heard is that this can be concentration sensitive. As you get to high concentrations, as is typical, right, you may be fouling the filter and reducing the performance. The other challenge of this today is that there's really no scale-up option here. And so if you're getting above a few liters of volume process, then really this is probably not a tangible solution for scale-up. So looking at this, while across the board it does well, it's not going to do so well if you need to scale up your process today. Thank you for the feedback that the audio is echoing a lot. I'll see what I can do about that. And James, if there's any guidance you have, I would appreciate it. The next one is tangential flow filtration. This could be TFF or ATF. It could be hollow fiber or flat sheet. And some really nice pros here. It's a closed system. It can be automated. It can be really nice buffer and media exchange. Scale down quite well and scale up as well. And There are some minor limitations here and maybe some considerations. We'll go into more detail on performance. One of the considerations here is processing time. While there's great solutions, this is an example from a vendor today. There are great solutions across the process feed volumes. There isn't, as you scale on a particular device, for example, this 0.02 meter squared device, as you increase the volume, you increase the processing time pretty linearly and pretty dramatically. And that means that if you go from 1 to 2 liters, you're doubling your time. 2 to 3 liters, you're tripling your, or 1 to 2 liters, 1 to 3 liters, I should say, you're tripling your time. But then if you go to the next stage, you may no longer have the same processing time. And if that is critical to you in your process to have an equivalent processing time on scale up, that can be a bit of a challenge. But across the board, for the most part, TFF does quite well. In fact, we saw that in the poll earlier. I think we had maybe 20% of folks were looking at TFF. Taking a closer look at this, one of the challenges of TFF is that as the cell concentration comes up, you can reduce performance. You can get filter fouling and reduce your flux. And this also can increase the shear on the cells, which is why Nimble, for example, came out and recommended that they only concentrate between 5 and 10x on their TFF device. Similarly, when you're looking at washing efficiencies, to achieve high washing efficiencies, you need fairly large amounts of dia volumes, which can increase your buffers, like cryopreservation buffers, which can be quite expensive, as well as increase your processing time. So even if we concentrate 5x or 10x ahead of time, we still have fairly large dia volumes to get our washing efficiencies, which drives both cost and time. And lastly, hold-up volume and hold-up loss potential. From an existing vendor, this graph represents their hold-up volume as a percentage of their recommended working volume. You can see across these different scales, it ranges from 15 to 50%, which means there's a major necessity to chase or flush the cells, which can also drive a dilution of your final concentration, reducing the optimization of your final concentration for whatever next process that there is. Overall, tangential flow filtration meets the performance very nicely. While it's maybe not the easiest thing to enter with and may have some performance challenges, it goes well across the board for cell handling. Now, though, diving into tubular bowl centrifugation. And before I get too far into kind of looking at this as a technology, I want to share a little bit about how it works today. So we start on the left here. We have our inlet port where the cells can be fed in on the center line of the bowl. As they get introduced in the feed tube, they get fed into the bowl body, which is rotating. And if we go to step 2 here in the concentration step, as the feed is sent in, the cells separate out and stay along the bowl body edge, and the supernatant or clarified material solution comes out the top of the bowl. This allows for continuous centrifugation of a large volume at once. And on step 3, that feed can be chased by a buffer, which allows for washing of the concentrated cells in a smaller volume. And ultimately, then the cells can be resuspended in that wash buffer and discharged out of the device. And what we offer in terms of the system is equipment which can scale from 5 liters to greater than 500 liters with very short install times, single-use assemblies that come with this and have stock standards, and then automation, which enables near push-button, walk-away recipes and has very good repeatability. So if we compare Unifuge to TFF and look at processing time, for example, we can see here in the dashed lines the 3 products, for example, the UF Mini Pilot and U2K. And there's a couple of key callouts here and how they differentiate from TFF. One is a lower minimum processing time. So even as you get to small volumes, we can bring the processing time quite low, and we can keep it quite low throughout, even as we scale up. This means faster runs and higher throughput at your process development time, and faster runs when you scale up. When we have that lower overall processing time, even at 500 liters, we can be processing that material in less than 2 hours. And because these have very broad windows of processing, you have fewer consumables to worry about, and you can have simple recipes and simple critical process parameters. And if we look at concentration and washing, one of the benefits of the unifuge is that it's not sensitive to the concentration that comes in. As the concentration comes in, we're able to continue to concentrate that in any given device, such that you can achieve concentrations that are substantially higher than TFF or ATF because of their sensitivities to fouling. And the same can be true for the dye volumes. Because we kind of concentrate the cells all in a single volume, you can then have basically a near constant flush volume or wash volume to achieve that 99%, regardless of the number of cells or the amount of concentrating you do of your cells. Maybe lastly, looking at it from a holdup volume percentage, we talked about the TFF having 15 to 50% of the working volume being held up. In the unifuge systems, that is as low as 0.5% to 5%. This means you can increase your cell densities and reduce your buffer utilization overall in order to get that maximum return. So then if you look at scaling, what do we have in terms of an offering? What does the unifuge provide? We have the UF Mini equipment, which can flow from between 100 milliliters a minute up to 1 liter a minute, and can fit on your bench top. We have the Unifuge Pilot, which can flow from around 1 liter a minute to 4 liters a minute. And we have the U2K to a very large commercial scale, flowing from 4 to 10 plus liters a minute. So it's great that we have that in terms of scale up, and we show that we have the equipment across the scale. But really the question is, do we get the performance across the scale? And so one of the things we wanted to look at is, can we look at a modeled, empirically validated scalability? That all starts with the bowl design. We've worked very hard to have a bowl design that has critical geometric elements that help scale the performance. One of the other benefits we have in terms of having robust scale-up is that we have very few critical process parameters to tune. It really boils down to two key parameters: your feed flow rate and your relative centrifugal force, RCF, or what often is called the G-force. And taking those two things, we can simplify our predicted scaling using what's called a sigma factor. The sigma factor is a function of your RCF, your centrifugal force, and the geometry of the bowl, which we carefully control. And when we put that together with the Q, or the flow rate, we can have predictable clarification cell recovery across scales through the sigma equivalence equation, which says that the flow of the first centrifuge divided by the sigma of the first centrifuge should equal the flow of the second centrifuge divided by the sigma of the second centrifuge. And when those are equal, you would anticipate having equivalent performance. And so we can apply that to our centrifuges, saying, as we go from the UF Micro to the UF Mini to the UF Pilot, we can look at those numbers and say, wow, we predict the performance of the scale up using the smaller scale. In this particular case, we're putting together we have a case study where we looked at flow rate ranges that had a 60-fold difference from 50 milliliters a minute to 3 liters a minute. Bowl volumes from 90 mils up to 1.8 mils, and some extremes in terms of relative centrifugal force. We looked across the Micro, the Mini, and the Pilot here. And what we found, utilizing the sigma scaling to predict that linearity, is that we were able to predict the clarification power. As we looked at Q over sigma of those 3 different devices across that test matrix, we could predict the turbidity reduction and the scalability of that turbidity reduction. In addition to that, we could predict a viable cell recovery yield. That, again, across all of the different sizes and flow rates, we could predict that with fairly good robustness. And what this means is, once you have this model for your system, you can predict out what your viable cell recovery and what your clarification will be as you scale up. What's really also promising is that the viability of the cells is maintained regardless of the performance, which means, unlike some other systems, as you increase or intensify the performance of these devices, you don't see substantial impacts or deleterious effects to meeting the CQA of viability. So overall, kind of looking at this from a pros and cons standpoint, The unifuge can give you the closed system, the automation, the buffer media exchange, the scale down and scale up, processing efficiency in terms of time, high washing efficiency. But are there limitations? And one of the greatest limitations that we ran into with end users is ease of entry. We know that the system is not in everybody's lab like a benchtop centrifuge, and so we would like to assist with that. We would love to ease the entry and access to this by doing a demo in a day. And it's not exactly a day, it's closer to 36 hours. So we'd love to do a rapid demo with you and provide that preliminary optimization of your process. And what that looks like is us bringing equipment on site. It could be a UF Mini or a UF Pilot with the consumable associated with that. We like to bring additional measurement tools for looking at turbidity and clarification efficiency, as well as cell recovery, in addition to what you already have in terms of your cell counter or other viability or other metrics. And of course, we'll bring an application specialist along to support that. In terms of timeline, we work with you with the pre-demo planning to plan out your tests and help you optimize those through our experience. Then day of or the day before, we can set up the equipment with you. Typically day of, we can do one or more process runs. I would say on average when we work with customers, we're able to use the material to process up to 3 or 4 different conditions. And then we can take down that equipment all in a single day. And what we'd love to do is then follow up with you with that data analysis. Of course, all along the way, you can have an expert along with you for your application support. One question is, does this work for your cell line? And just want to share with you that we've seen great performance across a number of different cell types, from iPSCs to TILs, stem cells and NK cells, and a number of other cell lines. So if you're wondering today about your cell line, we'd love to share that with you. And just a little bit of a deeper dive on a couple of existing case studies we have to give you a better taste and flavor of how this works and how this looks against some of the other technologies. First case study would be looking at iPSC media exchange and differentiating these cells. Again, this is available, I think, in the content today, but also on our website for more information. In this particular case, the end user was looking to differentiate their iPSCs and utilizing media exchanges to bring in and out critical cytokines to get the differentiation they wanted. Throughout their process and differentiation process, they had multiple media exchanges that each had a different volume, and that had to be very specific in terms of their concentration and the media exchange to achieve the differentiation they were looking for. Their control system they were using and their control process was gravity sedimentation. As we talked about, this is a manual process for them. They had limited volume control and had a major scale challenge for them that they perceived. Ultimately, when we were able to compare these two, the control here in blue versus the UF micro, which was being used, we had several points to evaluate the CQAs of the process, or of the product here. The first set of biomarkers, in both cases, at the midpoint on the left, the UF micro outperformed the control in terms of cells with the markers desired. And on the right, this was the endpoint differentiation. Again, the UF micro showed as good or better than the control in terms of viable cells with the desired markers. And when we looked at morphology at midpoint and endpoint, comparing the control to the UF micro, we can see that in both cases, the morphology looked equivalent. Ultimately, what they were looking for was final differentiated cell concentration. And again, the UF micro performed comparatively or better than the control. So overall, for them, it enabled this end user to have an automated closed system that allowed for repeatable media exchange, kept their aggregates intact, which was really important for them, and enabled that targeted differentiation to get that final concentration of their differentiated cell product. In this next case study, this particular end user working with PBS Biotech they were looking to have reproducible iPSC seed train expansion. And so in this case, they were looking to passage the cells with media exchange between every passage. And the control system here was a benchtop centrifuge. And as we talked about, this was an open process, manual handling, and had some scaling challenges. And both in 2D culture and in 3D culture, we passaged and then compared the performance between passaging with the control or passaging with the UF Mini. And we can see that the expansion was functionally equivalent across both of those. As well as when we looked at the morphology here, on the adherent cell culture, we saw some elongation. Both the control and the UF Mini were comparable, as well as in the 3D expansion, the aggregates showed comparability of morphology. And ultimately, the growth rates were fairly uniform across these, but more importantly, the pluripotency marker expression continued to stay healthy across all of the passages with the UF Mini. So for this case study, they were able to use an automated closed system, keep that reproducibility of passaging, and keep that pluripotency, which was so critical to this particular process. Lastly here, looking at high viable yield harvest with MSCs. This end user, again, was looking to seed the MSCs, do selection expansion, and then focus on the harvest step. And there they were comparing to a semi-automated membrane filtration. For them, they were having troubles with having cell loss in this process, prolonging the process and having too long of duration, and ultimately then a scaling challenge as they look to scale up their process. And so here on the left, we can look at viable cell recovery. The range of viable cell recovery they were getting with the membrane filtration is here in blue, from around 60 to 75%. And our very first shots on goal with the UF Mini, trial 1, trial 2, and trial 3, this was just basically in one day, and we were able to get 90% or above with very simple optimization. In addition to that, we were able to cut their processing time here on the right by about half to get a faster overall process. So for them, they were able to get a high recovery yield, a reproducible harvest, and then have efficient harvesting times. So as we wrap up for today, today in review, as we talked about early with these cell handling processes, picking the right technology can have important outcomes in terms of how you perform in the long term and how you perform in the short term, and how your scale down is predictable for your scale up. And that the unifuge systems, while they're recipe-driven and closed system, that gives a lot of performance, gives that predictable scale down and scale up, supports a number of cell lines, and gives you that straightforward early access to the critical process parameters, allowing you to have early definition of your critical quality attributes. Ultimately, confident scale up starts with better cell handling. And with that, we'd like to invite you to reach out to us. If you're interested in exploring with your process, you want to do proof of concept runs, you want to do process optimization, we would love to work with you. So please reach out and connect with us so we can work with you to have that quick demo. And I'll just say thank you very much for your time, and thank you, James. I look forward to answering the questions on that.
Thank you, Mark, for the excellent presentation. Before the Q&A begins, I'd like to remind our audience you can connect directly with Mark. His information is located on the side of your screen. We see a lot of great questions coming in. Thank you for that. Please keep submitting those via the Q&A box below me. We're going to address as many as we can, and the rest I'll pass along to Mark and the CARR Biosystems team to follow up offline. Our first question comes from Julia, who's asking, for organizations anticipating significant scale jumps in the next 12 to 24 months, what should they be doing now to set themselves up for right the first time scale up?
Thank you. Yeah, I think as we talked about through the presentation today early on, if you're looking to scale up, that's when you really want to have confidence that you can predict what your critical process parameters are and how they're going to impact your CQAs. And selecting a technology early that allows you to really define those critical process parameters and that impact can really make it so you move from having to optimize your process when you scale up to be able to optimize your process during the scale down. So we hope that you've seen that the Unifuge is something that can do that for you today, even if your process scale is small today. Your process scale of today can fit. We can develop those with you and move to process scale of tomorrow.
Excellent, thank you. Our next question comes from Ian asking, for those of us who aren't working with stem cells, what are some other cell types and applications where you've seen this approach work well?
Sure. So getting a little bit, as we talked about at the top, we have a number of applications where our technology is utilized. We see a lot of cellular agriculture, which kind of has the same demands to keep the cells healthy. That there's a lot of interest there and use cases there, but also in other clarification approaches. For example, when you want to clarify, for example, for MABs, that's an application we've seen, especially though for viral vector development, especially for lentiviral development, where clarification can be really critical. We've seen some good success there, as well as a number of other applications that are across life science. I guess I'll leave it at that.
Excellent, thank you. Our next question, given the tubular bowl of the Unifuge is also a centrifuge like disk stack centrifuges, why doesn't it experience or generate similar shear stress and cell death?
Oh, that's a very good question. Yeah, that's a very interesting question. So not to get too technical here, but inside of a disk stack centrifuge, there are lots of disks that are rotating through the fluid. They create a lot of viscous shear. Every surface creates a lot of viscous shear against the fluid. And all of that viscous shear is what drives that ultimate shear cell death. And in the Unifuge, in the tubular bowl, we don't have that same type of viscous shear because we don't have all of those disks interfacing with the cells as they're moving through. Instead, we accelerate the cells in the solution out to the edge. So when they are impacting on the edge, there is limited to no shear at that interface, which is why we can then see those results where we get high viability across a broad operating range.
Excellent, thank you. Our next question, you mentioned the 3 hardware, but 4 consumables being micro, mini, pilot, and U2K. What consumables pair with what hardware?
Oh, great question. So our UF Mini, which is our smallest device, can fit in most laboratories on the bench. That UF Mini equipment pairs both with our UF Micro and UF Mini consumable. As the name implies, the UF Micro gives you even more scale down than the UF Mini. And so really depending on what scale you're at and how small you want to start, we can either start with the Micro to get very, very small scale performance or go to the Mini for kind of a modest process development performance volume.
Excellent, thank you. Our next question is, how does the Unifuge platform maintain equivalent CPPs across micro, mini, pilot, and the U2K systems? And why does that matter for tech transfer and right the first time engineering runs?
Sure. So because we have only 2 critical process parameters, typically we're looking at the flow rate that you're feeding into the device and the centrifugal force that you select. That simplifies the overall kind of process development matrix, variable matrix, if you will. And that in and of itself makes typically the process development with some heuristic guidance, for example, from our specialists, fairly straightforward at a given scale. When I talked about the sigma factor, that is the tool that allows us to then scale across the different volumes. Once we have a good sense of what your scale and performance is at a given volume, let's say in a mini, UF Mini, then we can predict out the scalability to the pilot. And by landing either dead on or very close to optimal parameters, when we scale up, then allows for a lot more optimization instead of development at that large scale, which can then streamline, get you closer to that right first time when you do scale up.
Thank you. Our next question is from Roberto, asking if you could please elaborate on feed conditions that work toward continuous manufacturing.
So this is I can interpret this question many ways. So if I interpret it wrong, Roberto, I apologize. But in terms of, I did mention earlier that we have this, it's a continuous flow centrifuge. And in this particular case, continuous flow means we can flow in the centrifuge with the feed and continuously discharge the clarified supernatant or buffer that's coming out. In this sense, the cells do continue to pack inside or build up inside of the centrifuge bowl itself. And ultimately, if you do have enough cells or enough volume, you do need to discharge those cells out before you can continue to flow. So if, specifically to your question, Roberto, about continuous manufacturing, it really depends on what you're looking for in that framework, which process step you're trying to achieve, whether you're doing media exchange for perfusion or some other application. That would really drive kind of the definition of how this could fit into a continuous workflow and continuous manufacturing.
Perfect, thank you. Our next question, coming from Gia, is asking, what is the outlook for using single-use centrifugation in perfusion processes, and what strategies are available to control shear stress while maintaining stable long-term operation?
Yeah, that's a great question. Since I just mentioned perfusion, not something we talked about here necessarily, but yeah, when you're talking about perfusion, which is really just a prolonged media exchange, right? You're just bringing the medium out and bringing the cells back in, retaining those cells. These types of centrifuges can be used for that. We have seen that use case from some of our end users today, where you can bring the cells out, discharge a large portion of the spent medium, for example, and then bring the cells back into the reactor. I'm not sure I'm answering all of the elements there, but let me just see if I can look. Oh, and then maintaining the shear stress. So because regardless of kind of how many cells we have in the device, the shear stress functionally remains the same. Once the cells are in the device, they're not experiencing any additional shear. In fact, the majority of the shear may come from pumping in and out of the device, which doesn't happen that frequently. The cells pass in once and then pass out once. So it is able to maintain that viability over a longer period of time than, say, some other processes where the shear stress is prolonged and repeated.
Excellent. I think we have time for one or two more questions here. This next one comes from Sarah, asking, does the UF Mini allow for temperature control on the centrifuge bowl, or is the temperature dictated by the input materials?
Great question. And we actually have a couple of different options here, depending on what your needs are. On the mini scale, when you're talking about in the laboratory, typically the processing volumes are fairly small, and we don't have any temperature control there today. However, if you were on the device, I should say, exactly as you kind of mentioned in your question, you can dictate that or help assist in the temperature control by either pre-cooling your feed or utilizing feed buffers to help cool the bowl. Additionally, on that small scale, you can operate these in cold rooms or refrigerators if needs be, where you can induce that temperature control. As we get to the larger scales, we do have options where on the Unifuge Pilot and on the U2K, where the outer bowl casing can be cooled or chilled, which can assist then in the overall temperature control for your process.
Thank you. We have time for one more question here. Many teams struggle with process variability as they scale from development into GMP. From your perspective, what are the most common pitfalls in harvest and media exchange that lead to inconsistent CQAs?
Yeah, I think that's a great question. I think the one that I've seen most as we've worked with end users is kind of all the way going back to that slide earlier, we talk about that CMC strategy, and that we as development teams often get in this cadence of we've got to do it as fast as possible. And typically as fast as possible means turn to the closest piece of equipment that we already have. And that can be a bench centrifuge, for example. And we kind of saw that in the poll today. And what we've seen is that, then once you've developed this process around a bench centrifuge, where do you go with it? What do you do with it? How do you scale it? And those kind of forward-looking questions are forgot about in the now. And so part of our discussion today was, to hopefully help people think about what technology can you select today? What can you implement with ease today that makes your future more successful and reduces the pressure on the tomorrow as you scale up and tech transfer into commercial manufacturing?
All right, thank you so much, Mark. That is going to be all the time that we have left for today. I just want to assure the audience, don't worry. If we didn't get to your question, I'm passing them along to our presenters to follow up offline. I'd like to thank Mark and CARR Biosystems for providing their expertise today, and you, the audience. Appreciate you being with us and hope to see you again soon. Have a wonderful rest of your day.
Thank you.